muscle pain, encephalopathy, brain dysfunction, lactic acidosis. I already described that in strokes. I don't think I have to describe that. But that syndrome is caused specifically by one mutation in the mitochondrial chromosome, the A3243. Changing a A into a G makes that mutation. So that had been long recognized to cause this well-recognized syndrome that was usually seen in adults or older kids. And later, much later, quite recently, in fact, in 2004, it was recognized that this same mutation caused autism. But I want to show you the level of the evidence for this mutation causing it. Remember I told you that the mitochondria are inherited from mom. So what the group here, Sal DeMaro's group at Columbia, they found mothers of 4 patients who had autism. The mothers carried this mutation. They could find some copies of this mutation. The mother had lots of normal copies, but she also had some mutant copies. Now, the mothers, 2 of the kids who had autism had the same mutation you saw in mom. So they were actually able to see it. But 2 of the kids, they could not find the mutation that was in mom. Again, we know that the mitochondrial mutants come from mom. The abundance might have been too low to find, but they even looked in 5 different tissues and they couldn't see it. So it is extremely hard with current technology to be able to see, where am I going? To be able to see these mitochondrial DNA mutations. But remember I had told you that most of the mitochondrial genes are not carried on the mitochondrial DNA. Most of them are carried on those classic chromosomes that I showed you in the beginning, where you get a copy from mom and a copy from dad. That's true of even the respiratory complexes, those core components, and there probably are over 1,000 to 2,000 genes that are required to build a mitochondrion itself. So that takes us to the point of discovering the genes that are involved in autism. And I'm going to hit 3 areas that have been very prominent in the newspapers recently. One is this idea, One is this idea of associated genes, and that's genome-wide association. I'm going to try to explain that in a second. And one is the idea of SNPs, single nucleotide polymorphisms, and the notion of missing heritability, and then moving on to resequencing and the discovery of rare variants. I'm going to try to take them all on in one fell swoop because I don't want to digress into the details of any of them. But I'm going to give you a summary of the state of using these genome-wide association tests, which only became feasible about 3 years ago. And the old way an association test would be done is you would take one gene, and you would look at that one gene, and you would look for changes in the sequence of that gene in people with autism compared to people who don't have autism. With the advent of these gene chips, you could suddenly do that same experiment, but looking at all of the genes, looking at millions of spots on the genome in individuals with autism and individuals who don't have autism, and looking for a statistically significant peak that comes up in one of them. Now the problem, and I'm saying a problem, you won't usually hear a geneticist say it this clearly, because it's been agreed that we will say it has been a success, okay? Because hundreds of millions of dollars were spent on this. Basically, the problem was that to have the statistical power to carry out that kind of a study, you had to use variants. I'm going to just use simply calling the sequence had to be red or blue. There had to be enough reds and blues mixed in there in the population. The frequency of the rare allele had to be high enough that you'd get some information on that spot. If everybody was blue, there's no information. You're not doing genetics. You're not learning anything. You've got to have a mix, some reds and some blues. Well, it turns out that in order to reach the level that you can do the study meaningfully, the mutant has to be present at about a 5% or more level. So those are very, very common variants in the population, and that led to the theory. It's almost a circular or a tautology that, in fact, all of these common diseases would be caused by mutations that themselves were very common in the population. Now, in a sense, that had to be true or you couldn't do the study. if they weren't going to be common, if they were going to be rare, if everyone was going to be different, you'd never be able to find them using the technology that was available at the time. So that led to this common disease are caused by common variants hypothesis. And as I said, hundreds of millions of dollars were done making these association studies, and the driver always was, we need to get a bigger study. We need to get a bigger study. We need to get a bigger study. And this was done over and over again, and I would say by the middle of 2009, it was basically recognized that this is a waste of time and it's a failure. What we've accounted for is maybe 2% of that heritability that I told you was there. If you're very generous and allow them to wave their hands all kinds of ways, maybe you could say they account for 10% of the variability that's there. I just want to give you an example of that. One is in diabetes, a very, very easy disease to come by. It's not a rare disease to find. Finding about 7 or 8 variants that contribute to diabetes. You might think that's very meaningful. That's viewed as being a success. They discovered these genes that contribute to diabetes. But the amount of variation that they contribute is so small that to account for the heritability of the disease, there would have to be 1,500 such genes inherited together to make the disease. So again, if you're pointing everywhere, you're not pointing anywhere. You're not learning anything. For a simple trait, you might say, oh, you can't really diagnose that well enough. A very simple trait, height. If you take height and you look at the SNP, the common variants that have been found associated with height, it would take 93,000 variants to describe what we recognize to be the 80% heritability of height. So basically, there's essentially no information contributed by a SNP. And it's virtually certain in a lot of the modeling that's been done that to the extent that there are real SNP associations, what they're really doing is they're artificially associated with a rare genetic variant that's making the disease. No one's ever been able to go in and sequence near one of these SNPs and find the mutation that makes the disease. We care about the genes that make the disease. So again, what this has brought us back to after a painful excursion is we're back to the rare variants again. We now recognize that the common diseases, in fact, are caused by the same kind of rare variants that I told you we wanted to study because they were more simply approached. So we then move on to looking at the rare variants that we've been studying here at UCI. And these now have the name of copy number variants. That's where it's not changes specifically in the sequence of the DNA, but a very surprising finding has recently come about that we have blocks of DNA where they've expanded and copied themselves for millions of bases for reasons that we still don't understand. Let me show you one example that goes back to a paper that we published in 2003. And this was the finding of a 15q inverted duplication in autism. And this is a cartoon of chromosome 15. And the kids who have this disorder get one normal chromosome 15 from dad, one normal chromosome 15 from mom, but in the egg, while this was happening, a little block of that chromosome 15 duplicated itself. And sometimes it duplicated itself and stayed right here, making an abnormal chromosome that had 2 copies of a big block of DNA. But more commonly, it duplicated itself and then duplicated itself again and hopped out and became its own little miniature marker chromosome. We call it a marker chromosome that has this 15q region on it. So the kid has 4 copies of that block of the DNA. In fact, this is still the most common chromosomal abnormality that's found in autism. And what we found is that in the kids who had this, we saw the signature, the biochemical signature that we had seen in mitochondrial diseases. In addition to the autism, they had hypotonia, weak tone, ragdoll kind of tone. They had that lactic acidosis, these other biochemical signs. The mitochondria were sensing that they weren't making enough energy. They were proliferating. We could measure defects in the complexes, but the mitochondrial DNA was normal. Presumably, the abnormal dose of these genes here was contributing to this abnormal mitochondrial function. So the question was, is the line, the abnormal chromosome, leads to these biochemical findings